Hi, in this research paper, I'm going to be talking about a paper that talks about an inulin gel based oral immunotherapy that remodels the small intestinal microbiome and as a result suppresses food allergies. This research paper was published in 2024. Let's get started. Before we consider talking about the paper, I thought we would talk about a little bit of the background. But currently, the global population statistically has over 30 to 40% with at least one allergy. Furthermore, food allergies specifically have seen an increase since the 1960s from 3% in the populace to over 7%, which is more than a double increase. And furthermore, developed countries have higher percentage prevalence of allergies versus developing countries. And before we get into the real details of the research, I thought I'd talk about the question that the authors were trying to answer. The authors asked whether engineering an inulin gel with an allergen can restore a healthy small intestinal microbiome, suppress pathological immune responses, and lead to sustained protection against food allergens. And that sounds like a lot of jargon, but I think we should talk a little bit about what inulin in itself is. Inulin is a polysaccharide that is derived from fructose, and due to the structure of inulin, it is undigestible in the human body, which allows it for it to be an excellent prebiotic. And it's undigestible because of the beta 2 to 1 bonds on that fructose chain, as you can see. So, inulin can serve as a way to get things in the system without it getting fully digested. So, the authors created and administered an inulin allergen gel in multiple mouse models, comparing its effects on immune regulation and gut bacteria. And their idea is that if they can prove it to work in mice, then they can also get it to work in humans. And they did this by measuring the allergic responses between the mice and analyzing T cell subsets and profiling the small intestinal microbiome and metabolites. And we'll get into all of that. Before we talk about our research, I thought I'd delve into the prior history of immunotherapies for mitigating allergies in the field in general. And I should make the distinction that while our research talks about an inulin gel based immunotherapy, it is important to understand that it also falls under oral immunotherapies because it is administered orally. That said, oral immunotherapies of different kinds have actually been tested before, and one of them is actually approved, Palforzia, which is a peanut um, allergy medicine, but a lot of oral immunotherapies currently have a lot of problems. They don't work for some people, and other people they cause a lot of gastric discomfort, and it also has increased the risk of anaphylaxis in some people. And furthermore, they don't have any guaranteed lasting tolerance. Research shows that daily dosing must continue. There's other therapies too, such as sublingual immunotherapy and epicutaneous immunotherapy, but those all have their pros and cons as well, and they don't all work very well. The efficacy is modest, and they stop working if you stop taking them. So these histories of different immunotherapies have suggested that this is a very developing field at the moment, and we need to make better therapies to deal with allergies. And some of the therapies include strategically targeting the microbiome of different organs in the human body. And I actually bolded the microbiome modulation because I wanted to use that to dive into our research, because the microbiome has a lot to do with allergy response, as we will discover. I think it's important to understand why the microbiome plays such a strong role in allergies. It's going to help us understand our paper better. So, in the early life, the microbiome is very important because it trains the immune system. The beneficial bacteria that is found in a good microbiome, those bacteria's wastes and metabolites influence the maturity of immune cells. The specific bacterial families, such as Clostridia or Bacteroides, they promote regulatory T cells, and by doing so, and also releasing metabolites such as short chain fatty acids, these help suppress IgE mediated allergic reactions. Now, as a reminder, IgE stands for immunoglobulin E, and you can see in the figure that I show there, IgE is what activates our mast cells and sensitizes them. And if an allergen causes our immune system to be sensitized like that, that's how we release histamine and cause an immune system response that can lead to anaphylaxis and all that. And furthermore, I should say, because of the importance of the microbiome, antibiotics and diets which affect the microbiome also play a major role in allergy development, and it can cause our immune system to have more inflammatory responses to foreign substances. So, with all of that, the restoration of a balanced microbiome is very important. So, this kind of begs the idea that if you can mitigate allergies, especially food allergies, the most likely way to do it would be kind of a 1-2 combo of immunotherapy and reducing a good gut microbiome, or generally a microbiome, and this would help boost efficacy and increase long-term immune system tolerance. And a lot of these bacteria are very important. So, I thought I'd start diving into the article. The article seemed to want to answer a lot of questions, but the three most important questions that the scientists wanted to answer is: can we test an inulin based gel in mice as a vector to give allergens as an oral immunotherapy? And would it work better and safer due to the 1-2 combo that I discussed? The second question is: would this work better than regular oral immunotherapy because the inulin approach would be safer and the targeting of the gut microbiome would increase efficacy? And the third question is: compared to traditional oral immunotherapy, does inulin oral immunotherapy reduce allergies, work strong over time, and establish allergen specific oral tolerance? All right, so I thought I'd talk about the methodology of the paper. So, to test the questions that the scientists had, they did this on mice. And these mice were sensitized to OVA, or oval albumin. You might think albumin and think eggs. It's like a compound that's in eggs. And the way they made mice basically allergic to them is by they used an aluminium adjuvanted model. And the way I'd explain it is it's a vector to basically induce allergies. It's like making a vaccine, but you're inducing an allergy. So they injected mice with this OVA vaccine on day 0 and day 14. And then 28 days later, these mice were split into groups and then treated with different oral immunotherapy regimens. And they had multiple groups. They had a standard OVA oral immunotherapy where they would give the oral administration of free oval albumin. They had a group of inulin and OVA, which is the OVA that was mixed with inulin but not gelled. And then they had an inulin gel OVA oral immunotherapy, which is the OVA that was incorporated into a structured inulin gel formulation. And then they had some other group, which was a guanosine OIT, where they were trying to assess immune modulating effects of specific gut metabolites. It's to drive their results better. And then they had two control groups, which are the native non non-allergic mice, and then they had the OVA sensitized group. Continuing with the methodology, on day 48, they tested fecal samples of the mice through 16S rRNA, and then they sorted the bacteria that they saw in the fecal samples into protective versus inflammatory species using Motor. On day 49, they had an allergen challenge, and further methodology included doing data derived from the metabolomics, or just checking the metabolites of the mice. And they also did flow cytometry to measure the counts of regulatory T cells, and also a lot of cytokines such as IL-4, IL-13, IL-10. And they also checked the mast cell degranulation, which is important because the mast cell degranulation is a marker of anaphylaxis. The cytokines promote or suppress IgE production, and the FOX3 regulatory T cells, they are important because they suppress allergic responses. And furthermore, they also did gut barrier integrity tests, and this is done to evaluate whether inulin gel improves the gut barrier function, which helps prevent food allergens from excessively activating the immune system. And mice were also given this thing called FITC dextran, and that's some marker, a fluorescent marker that leaks into the bloodstream if the gut barrier is impaired. And then they did a lot of statistical analysis via ANOVA and Mann-Whitney U tests. All right, so I think we can start talking about results. But I think it's important to understand what the scientists were trying to test for. In this page, I'm going to be talking about kind of the phenotypes that they are observed between the various groups. And these phenotypes are stuff such as like anaphylaxis scores and stuff like diarrhea and body temperature, because these are all symptoms that would be invoked if you gave allergies and had an allergic response to it. And so you can see on the graph, and all of these graphs will have these groups, but you can see on the first graph, C, where you have four groups, one being the naive group, one being the OVA group, which is just the allergy in itself, one being a PBS group, which is a negative control group, and the red being the inulin gel OVA group. So let's first look at this one. In this test, they were trying to see the body temperature drop or do a hypothermia test to see if the therapy is working. And as we can see, the inulin gel group actually performed rather similar to the naive or native group, basically, which suggests that it worked. And we can also see similarly in graph E, where the occurrence of diarrhea when they challenged the mice was the lowest in the inulin gel group, and much higher in the negative control, and also the just the OVA group. And we can also see they did an aggregate score of the diarrhea on group F, and the inulin gel group is actually very favorable. And a bigger statistic I want to emphasize is that the inulin gel group prevented mortality in 100% of mice, compared to 19% and 31% in the OVA and PBS groups. And I should remind you that the PBS group is a negative control group. In these figures, we talk more about the IgE values, and as you can see, the IgE value in the inulin gel OVA group is the lowest compared to the negative control and the OVA group. Although not by much, but it is still statistically lower. And so that suggests that the inulin gel was working. In figure J, we can see the MMCP1, which I'll talk about in another second. And in figure K, we can see the mean mast cells being the closest to the original naive group. And I should emphasize that that's a important thing, because the mean mast cells being closest to the original dictate that the inulin gel was keeping the allergies from attacking and causing an allergic response. And also the MMCP1, as a reminder, is the mast cell degranulation, which is a direct indicator of anaphylaxis, and it is definitely the lowest amongst the OVA, the positive test, and the PBS group. And we also see in figure B, which is to test the FITC dextran level, the inulin gel group seemed to be the lowest. They also did further tests, and these are more longer term tests. And what the scientists were trying to see here is that if you stop giving the oral immunotherapy, the inulin gel immunotherapy basically, will the mice still remain free from the allergic consequences? And as we can see in the longer term test, the inulin gel group actually had a very minor change in body weight, which means that they weren't struck so hard with the allergy. And the anaphylactic score in the inulin gel group is much closer to 0, which is a good thing. And in D also, we see that the diarrhea occurrence in the inulin gel group is close to 25-30%, and so 60% of the mice actually remained free from diarrhea. And in line with this, mice that were also treated with the inulin gel, they maintained a long-lasting immune tolerances after stopping the oral immunotherapy. And even on day 79, mice still had significantly increased frequencies of FOXP3 and the regulatory T cells, which all indicate good allergy mitigation. And they also showed reduced frequency of the IL-4 cytokines and all that, which are inflammatory immune responses. And long term, the inulin gel OVA therapy really proved that even after you stop it, you reduce anaphylactic risk, diarrhea, and body weight changes overall. They also did a fourth test, which was a torture test, I would say, to put it in simple terms. And again, the inulin gel allowed mice to stay about 60% diarrhea free, and the anaphylactic score was good as well. I also wanted to highlight when they looked at the ileal contents off of the fecal samples. Look at figure F. In figure F, we can see those two arrows. One is the inulin gel and OVA group, and one is the naive group. And figure F shows the bacterial compositions of the ileal contents of the mice, and it's hard to understand this in a easy way, but I just want you to notice the similarity between the inulin gel OVA group and the naive group, which is the first group, and also look at the difference between the PBS group, the OVA group, and the inulin OVA group. You can specifically see that in the PBS group and the OVA group, they look completely different compared to the first and last one, the ones with the arrows, which suggests that the inulin gel OVA therapy maintained a good microbiome in the mice. And overall, the scientists were very successful with the OVA tests, that they also put it into different models, allergy models, in the mice by testing a peanut allergy model and a cow's milk allergy model. And as we can see in figure B, where they tested cow's milk, they tested casein, which is a protein in milk. The inulin gel casein group had a super low anaphylactic score, and the casein specific IgE. So they used a similar testing methodology and all that. They almost did a whole other experiment in itself to prove that their inulin gel immunotherapy works. They saw that the IgE value is the lowest, which suggests that the immune system was not showcasing a allergic response. And in further figures such as E and F, we can also see where they tested the peanuts and did a peanut inulin gel, and we can see that the anaphylactic score was super low when they tested it with the inulin gel, and also the body temperature drop is close to 0 in figure F, even at day 80. So all of these suggest that inulin gel as an oral immunotherapy combined with the allergen work really, really well to prevent allergic responses. All right, so some discussions and conclusions. I would say that the results were a resounding success, and they proved that an inulin gel-based therapy can induce long-lasting immune tolerance to allergies, and it also helps shape the intestinal microbiome. The scientists proved that their model worked on other allergy models, the peanuts and the cow's milk. And the scientists also highlighted that this was a simple formulation of the allergen and the inulin gel, and they remarked that by the FDA, this is a rather safe formulation. So this would posit that if it worked so well in mice, then it also will work very well in humans. And that leads us. So I thought we would talk about further questions, but based off of the research that I discussed today, I would ask, what other allergy models can we use the inulin allergen gel platform on to test? What implications could result when we move this into a testbed for human allergy tolerance with human clinical and critical trials? What is the most critical factor in achieving immune tolerance? Would it be the microbiome changes or the regulatory T cells induction or something else? How would dosing schedule and allergy testing schedule with a potential treatment like this go? Would you try to induce anaphylaxis in people? It raises some ethical questions. And with research showing that the gut microbiome variability is present in humans, especially influenced by their genetics and exosomes, how would this play into the efficacy of such treatments? These are my references, and thank you so much for your time.